Dinoflagellates are one of the major components of marine phytoplankton community, which during favorable condition proliferate at very high cell density, resulting to what is known as microalgal blooms. So most blooms are actually beneficial since they form the base of the food chain, serving as essential food to zooplankton, filter feeding shellfish, and fish larvae. However, some forms harmful algal bloom, such as the genus Carenia in the family Carenaceae. This group of dinoflagellates are widely known for its ability to form harmful red tides in many countries, such as China, Japan, and USA, with devastating impacts on fisheries, coastal ecosystem, and public health. In the Philippines, Carenia mikimotoi is the only known species of Carenia, and its occurrence has only been reported in Bolinao, Pangasinan. So, the presence of Carenia mikimotoi and other related species in other coastal areas of the country remains to be determined and taxonomically verified, since Carenia exhibit high morphological plasticity and their cells could be easily deformed when fixed during routine phytoplankton monitoring. So my main research question is, what species of Carenia are present in other coastal waters of the Philippines? So to answer this, water samples were collected in Manila Bay and Subic Bay, and live cells showing typical swimming behavior of a Carenia species were isolated and eventually cultured for LM and SEM observation, as well as molecular and pigment analysis. Result of our taxonomic identification showed the presence of Carenia mikimotoi in Manila Bay, and the first report of Carenia papillonacea from Subic Bay in our country, and even in Southeast Asia, based on my literature review. So identification was based on the characterization of their morphology and determination of their phylogenetic positions as inferred from LSU rDNA and ITS region. So mainly, the cell shape, position of the nucleus, shape of the apical structure complex, and the shape of the chloroplast, and the type of pyrenoid, the rich pool of carotenoids, are taxonomically informative characters used to identify the Carenia isolates. So to sum up, our study reported 2 species of Carenia from Manila Bay and Subic Bay, of which Carenia papillonacea has never been reported in our country before. So the 2 species were identified by detailed morphological characterization, phylogenetic, and pigment analysis. So since the occurrence of Carenia species has been extended to 2 more coastal areas of the Philippines, further research needs to be conducted to better understand the diversity and distribution of these potentially harmful dinoflagellates in other coastal areas of the country. Thank you.